So our speaker tonight is Yuhai Tu. And let me say, he's an outstanding roommate. He's a fabulous cook. He's a demon hiker. But I've also been reading his articles for many years, and they're full of insight. And this time, he's going to tell us an amazing adventure story. The incredible discovery that single cells are smart. Single cells have behavior. They make decisions. Your brain is billions and zillions of cells. One single cell is also very smart, makes decisions, performs calculations. Nobody realized that until the story that you're about to hear tonight. So finally, good roommate, good scientist. Yuhai is a fantastic citizen. He has been chairing our professional society's arm of biological physics, and as I sort of step into the apprentice role of that job myself, I'm realizing what a huge amount of volunteer work Yuhai did and how well you did it. So thank you very much, Yuhai.
Thanks. That's very generous. So before I start my talk, I'd like to say a few words about the center myself from my personal perspective. I came to the center first time, 1991, when I was just graduating from my graduate school, getting my PhD. So if you remember 1991, the economy was not so great. So my friends in physics, a lot of my friends in physics has decided to pursue something else, like in finance, in Wall Street. So anyway, at that time I was sort of wavering a little bit, not too much. But then my advisor said, you should come here, right? You'll have fun. So I came, 91. Oh my God, I did have a lot of fun. I think, well, doing physics is not too bad. At least you can come to Aspen, spend the summer, and have a lot of fun, right? So that was actually made a huge impact in me. It's like, doing physics can be cool, can be very interesting. Another thing I want to say is actually with respect to this type of public lecture. The same year, 91, I'm going to show you a picture. Yves Couder is somebody I really respect. Some of his work inspired my PhD work. And in that year, 91, he was also in the program that I was in. And he gave the public lecture on something called the phyllotaxis. This is how plants grow these little seeds, little buds, and they form this wonderful pattern. For example, if you look at the sunflower, it actually formed a spiral, and one way and the other way, clockwise way. And you look at the number of arms in each way, they actually are two consecutive numbers in the famous Fibonacci series. This is the Fibonacci series, right? 2, 3, 2 plus 3 equals 5, 3 plus 5 is 8, and so on and so forth. And you look at this pine, look at this sunflower, it formed this beautiful pattern, two consecutive numbers, and the ratio is roughly the golden mean. So that also gave me, and I was in the audience as one of the many inspired young physicists, say, this is really cool. Physicists can make contribution to other than physical objects, but also living things. So I think I didn't know at that time, because I only started doing biophysics in the last 10, 15 years. But I think that made a lasting impression on me that actually physics is about law of nature, and that does include living things. So today I'm going to tell you something that is along that same line, but in the spirit of Yves. And by the way, Yves passed away a few months ago. He was a very distinguished physicist. And this sort of my talk, when they first asked me whether I want to give a public lecture, I hesitated like a nanosecond. Then I remembered Yves. Then I said I should do it. And here I am. So, okay. So as advertised, my talk is very formally called Information Processing in Single Cells. What and how does E. coli compute? Very boring, very technical kind of thing. But what really I'm going to tell you about is how E. coli find its food. Okay. Oh, let me tell you a little bit about the E. coli, what E. coli is. E. coli is just the simplest and most interesting, I think, single-cell organism. It's roughly a rod-shaped cell body, about 2 micrometers long. What is 2 micrometer? It's about 1/10, 1/12 of a human hair diameter. Very, very small. And the diameter of itself is about 1 micron, half of the length. On its body, it has this very long, what people call flagella, 10 micrometer, actually longer, 5 times as long as the cell body itself, that propel its motion. It's like and the bottom of this flagella are these little tiny nanomotor rotating around at a speed of 300 hertz, maximum speed. 300 hertz is every second, 300 rounds per second, right? Driving it to move. It reproduces about every 20 minutes if the condition is right, if they have enough food. So that's very important to find food in the right amount of time. Otherwise, you will either starve to death or wouldn't be able to reproduce. Okay? So now it also moves, as I said, propelled by these flagella. It has around 4 to 6 of them. And the swimming speed actually is amazing. It's 20 to 30 micrometer per second. Give you an idea how fast that is. If you take the body length, which is 2 micrometer, this is about 1 second. It actually swims about 10 to 15 body lengths. If you translate the body length to a human body length, that is, so the fastest human is it's Usain Bolt, of course, right? So he actually runs about, 1 second, 5 body lengths. E. coli outswim Usain Bolt. Okay, it just says it's very, very fast. It needs to move fast because it needs to find that food, right? Now, is moving fast enough? Well, before I tell you that story, let me tell you how it actually moves. So in 1972, Howard Berg, pictured here, is a professor at Harvard, developed a tracking microscope. He was able to develop a microscope that actually tracks individual moving cell. So he can look at the trajectory of how it actually moves. Under that microscope, he was able to observe this kind of motion. So indeed, it swims around, but once in a while, it changes direction, right? So it doesn't just always go straight, it changes direction. And upon further detailed investigation, Berg and Brown found that there actually really only 2 modes of motion. Either when the motor, remember the nanomotor underneath the flagella, is rotating counterclockwise, if you look from outside in. And the 6 or 4 to 6 flagella will actually form a coherent bundle. Then it actually pushes the cell swimming forward. But once in a while, one of the motor, or 2 or a few of the motor, actually start rotating clockwise. So out of sync with the other guys. And then as a result, the bundle flies apart, and the cell just jiggling around a little bit. And then when they start to reform that bundle again, it's in a new direction. So as a result of this tumbling motion, the cell in the next time frame is actually moving in a different direction. And that direction seems to be independent of the previous direction. It's chosen basically randomly. Okay. So in physics, we call this random walk. So you can imagine this is the cell. It actually moves forward for a little bit, for about 1 second. And then at this tumbling event, it changes direction, choosing the random direction to move for another second, and then do it again and again, right? So E. coli's random walk referring to this type of motion, which follow actually the laws of physics. So physicists has long studied random walk, or in other words called Brownian motion. So I'll show you what do we mean by that. Actually, the Brownian motion, random walk, Brownian motion was discovered by a botanist, a biologist, right? Robert Brown. He was actually looking under a microscope of a small particle like pollen. And then he see very beautiful, what he described, a random walk motion, or what he called a bouncing motion of this little particle moving around randomly without any direction. And later on, this motion is called Brownian motion, right? And if you, and this is actually the simulation showing to the right, I'm going to show you in a minute. It was done by Phil over there, who introduced me in the last hour or so on demand. So what you're going to see is I start my particle all together, 50 of them in the middle, and I allow them to do random walk, and I see how far they can reach, right? What's the region of search? How far they can go. Now, if you look at this movie, it's going to start 1, 2. Oh, I have to type it. Okay. There it is. See, it all started in the middle. Now it started to spread out, right? As time goes on, it spread even further and further and further, right? So now if I start all the particles at the same place at time t equals 0, and I ask how far it will reach at time equals t, some future time t. It actually follows called diffusion equation. As a result, we can calculate this length scale, how far it will reach. And it goes like square root of d times time, t. T is time. What is this? The d? D is a quantity called the diffusion constant. Okay, so how many people has heard about diffusion and diffusion constant? Okay, good. The main thing to take home is that actually the distance you travel is not, suppose you have a constant speed, the distance you will travel will be linearly proportional to the time, right? If you go in the same direction, it will be the velocity times the time. And in this case, if you randomly move, the distance you're going to, the spread you're going to have, the distance you're going to travel, the net distance you're going to travel, only goes like t to the square root of t, slower. By this constant, the prefactor, the d times t is called the diffusion constant. Do anybody know who figured out what the d is? How do you calculate d? Okay, so it was Einstein. In 1905, he actually wrote 2 famous papers. One of them is about thermodynamic theory of diffusion. So he actually know about the phenomena of Brownian motion, see the particle moving around. He asked himself, what determines the d, the diffusion, right? Because everything else is given time, you measure the given time. So to determine the distance you travel, you just need to determine d. So the distance, so the diffusion constant was written here in this formula. Turns out in the physical system, only depends on the temperature of the environment. So Brownian motion, because the reason it's bouncing around is because there's a lot of other particles that bump into it, right? And so as a result, it actually moves around randomly because many other particles bump into it. And to describe how hard other particles bump into you, that description is given by temperature. So that's why the diffusion constant depends on temperature. What else does it depend on? It depends on how big the size is, right? So if your size is big, you probably move slower, right? So this formula basically tells you, and of course, if the media is very viscous, right? If you move in molasses versus moving water, you'll be moving in different speed. Okay, so basically Einstein had this formula. But in our case, actually, we are looking at an active particle, right? This particle has its own driving force, have its own motor. So it doesn't follow Einstein's relationship dependent on the size, or it depends on its own driving force. And the way it determines when to change direction. It's not passive. It's not passive bump by other guy and therefore move randomly. It actually, this time scale for it to change direction is determined by its internal decision making. So that's what Phil was talking about. The particle actually is pretty smart. It's actually used its motor and its brain, I'll tell you what E. coli's brain is about, to decide when to switch, when to change direction and when don't change direction. But if you just, on average, look at if in the absence of any outside information, it just moving around, just like a random particle, okay? Just like Brownian motion. And you can estimate the diffusion constant by looking at its speed. I told you about, it's about 25 micrometer per second. And the time for it to switch, right? How long does it run before it actually switch? From these 2 parameters, you actually can get the diffusion constant for E. coli. It's about 625 micrometers squared per second. That's a diffusion constant. Now, why do I tell you this? I'm telling you this is that I'm trying to ask myself this question. Is random search good enough to reach the goal? The goal is the food source, right? Suppose the food source is 1 millimeters away. How long would I take one lucky particle, one lucky E. coli to find it? So from this formula, diffusion equation, diffusion length, I know that the reach, how far it will reach, goes like square root of t times square root of the diffusion constant. I know it's 25. So I can answer the question, suppose the food source is 1 millimeter away. It will take 27 minutes to reach it. Remember I tell you the division time is about 20 minutes. So it really needs food to actually to divide and reproduce. It's too late. And furthermore, if it's 1 centimeter, that's even worse because it goes like square root of t. So 10 times more distance equals to 100 times longer. So this guy with this E. coli, poor E. coli, if it's only relying on diffusion, it would take 2 days. That would be way too late, right? Okay, so moving fast is actually not enough, right? So you have to be, what do you have to be? You have to be clever. You have to be smart. You have to find out where the source is. So it has to make good decisions, not just random ones, right? It is trying to make decision, changing direction, but it's random. It's throw a coin, you just try your luck. So you have to have better educated decision making. So how does that come about? And it turns out in Berg and Brown, they also did experiment in the gradient. So you put a food source here, or you put a repellent here. What they found is that E. coli actually does make good decision in the following sense. Remember I tell you in the absence of any information, it will just move and change direction every second, right? So it's random. But in the presence of the external gradient or external information, when it's going to the right direction, it actually moves more than 1 second, move 2 seconds. Then it switches. If it's moving in the wrong direction, instead of changing direction every 1 second, it changes 0.5 seconds. So as a result, you have a bias. Even though you're still random, you have a bias towards, your coin is actually not a 50-50 coin anymore. In the right direction, you have a high probability to prolong the run. And the wrong direction, you actually have a shorter run. You quickly decide to change direction, right? Okay, so I'm gonna ask a volunteer, pre-assigned volunteer, in this case, to play a game. This game, I think everybody played before. It's called warm and cold, hot and cold. Okay, so here is something you really like, right? So why don't you go there and turn around. Okay, don't look. Okay, now, all right, you know what it is, so I'm going to give you some hint. So the job is to try to find it. Cold, cold, warm, warm, warmer, 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 cold, warm, cold. Okay, I think you might. Okay, you cannot step on her. So yeah, that's basically what the idea. Okay, let's give me a round of applause. So what I'm trying to tell you is that I am the one who gave him information, right? I'm telling her that the direction is good. You should keep going. And if she's going away or overshoot, I say, oh, come back. I say, oh, it's getting colder now. So she comes back. So basically, E. coli follow the same game strategy. But the poor E. coli has to make that decision himself. There's not me. Nobody else is going to tell E. coli it's getting cold or hot or warm and so on and so forth. So E. coli has to make that decision. This guy, E. coli here, has to say, well, am I getting closer or I'm moving further away from the food source, right? in the presence of a gradient, because this guy does give out some hint. The carrot does give out some hint. It's a little smell particle, right? If you think about, that's how we smell things. We don't eat the smell. That's not nutritious, but the smell is a trait. Like a hint for us to find the food source, right? The same goes for the E. coli. The food does give out this little molecule, and the closer to the source, the higher the concentration. The further away from it, the smaller the concentration. So you form what we call a gradient, right? So it has to be able to compute that gradient, right? Because if you're going towards the food source, where then the concentration of this smell particle become higher and higher, then you know that's a good direction. If you go in the opposite direction, the smell particle, the smell molecule concentration become smaller and smaller, then you should change direction, right? Okay, so that's basically, I'm going to tell you how E. coli managed to do that. Okay, so obviously they did it. Under the microscope, they're actually doing that. So how does it do that? How does it, why it is actually smart enough? So the first question, in my title, I have two questions, what and how. What will I already tell you? It has to compute the gradient of the tracer particle or the smell particle. What it needs to do, what I'm going to tell you now is how does it actually manage to do that? Such a small thing, right? Okay, so the first answer is that how would you measure gradient? You measure gradient by, you can say, okay, I have two sensors, my left hand, right hand side, hand. I say, okay, is it hotter here or hotter here, right? If it's hotter here and colder here, then this is the right direction, right? So I say, okay, that I should go to my left, or vice versa, right? So let's see whether E. coli can use that strategy without even moving to do that measurement to decide which side is hotter. You can do such calculation. Basically, you know the gradient. So let's say 1 micromolar per millimeter, because very far away from the carrot is actually very, very small. The signal is actually very weak. But if the smell particle fall into what we call the sensor box, so E. coli does have these sensors sitting on the surface of its, I'll show you a little later, of the cell membrane, actually be able to sense these particles. But is it sensitive enough to actually sense it? The difference between the left hand and right hand, right? That's the question. Now, I know the gradient, which means that every 1 millimeter, the concentration changed by 1 micromolar, right? So I can compute the number of particle difference in this sensor box, which you can sense on the left, and in this sensor versus this sensor box. So I call this front and this back. So the number of particle falling into the front sensor box minus the particle in the back sensor box can be computed by looking at the gradient multiplied by the length of the cell body times the volume of the sensor box, right? If you do that computation, this is Avogadro constant and so on and so forth in a 1 meter cube, and then what you get is 0.4548. What does it mean is that on average, the difference is only about half a particle. What does half particle mean? It means that half of the time is zero, no difference. Half the time it only differs by 1 particle, 1 molecule. The E. coli, even though it's pretty smart, but it's just this difference is too small to detect. So you cannot stand still, and detect the difference. So what else can you do? You move around, right? Because it can move, as we know, as I told you. So basically, that's exactly what it does. You move around, and you can move pretty fast. In 1 second, it actually can move 25 micrometer, about 12 body lengths, right? So if 1 body length is not enough difference, maybe 12.5 body lengths will be enough, right? So if you multiply that 0.48 by 12.5, you get 6. So in this case, if I measure here, and I measure here, if the gradient is indeed 1 micromolar per 1 millimeter, the difference is about 6 molecules. I will tell you that the actual E. coli sensor is actually very, very sensitive. On the 6, it actually can detect. So by moving around and sensing different part of this gradient of this field of particle, molecule, tracer molecule, you may be able to detect it. It's above your threshold of detection, right? Okay, is the problem solved? Problem solved? Wait, no. Why? Because, yeah, so you can compare two different locations, and the difference large enough above your threshold, you can detect it. But so basically, you're doing the calculation that, okay, the current time, you measure the number of particles n of t, and you compare that with the previous measurement you measured, let's say 1 second ago, number of particles at t minus, the tau is 1 second or whatever, 2 seconds. Then this difference is above your threshold, then you detect it. Sounds good. But how does E. coli remember this previous measurement, right? I measured here. Now I'm measuring here. You have to remember what you have measured before. So you have to have a memory, right? To do this computation, you have to have a memory. So that's interesting. E. coli has a memory. I mean, memory is something that we think about, in higher animal, human.
The clock too?
No. The clock is, you need a rough clock. Because I said that the tumbling time, the time for it to run before you make a tumble, it's about 1 second. That's at the time scale, roughly the time scale. Yeah. Okay, so it is a memory. So memory is something we associate with our brain, or so on and so forth. So in this case, let me define what I mean by memory. Okay. So what is a memory? A memory is actually a record of a previous event, an event that happened before, right? When I grew up, I went to this city, and so on and so forth, right? I had this ice cream, and I really liked it. So you make a memory, and then you move on, right? But even when you're old, you still remember that ice cream tastes very, very good, right? A memory is only a representation of the event, but not the event itself. It's a memory, right? It's not actually you are tasting that ice cream at this very moment. You just remembered the ice cream from many, many years ago. And of course, memory should last longer than the event itself. If after you eat the ice cream, you immediately have no memory, doesn't recall, then that's not called a memory. The memory time at least have to much longer than the time scale that particular event happened. So these are what I think are necessary conditions for memory. We can talk about other higher order of memory, but this is my definition of memory. And I'm trying to convince you that E. coli does have this type of memory. The key is separation of time scale. The event happens in a short time, and whatever counts for the memory lasts longer, much longer than this time scale that this actual event happens. Okay. All right, before we do that, I said there's two necessary conditions to do this calculation. One is that you actually have to do the measurement itself, right? So you have to be able to measure at this very moment what it is, and then you put in the memory, and then do the next measurement. So let's first look at how does E. coli do the measurement? It actually does measurement through this, like I said, the receptor. What is a receptor? Receptor is a protein, a rod-shaped thing, right? Again, and I'll tell you the size of this. It's pretty small, about 20 nanometer, 20 angstrom here, 2 nanometers here, and the length is about 300 or so micrometer. Nanometer. What happened for this receptor, structure of the receptor? This is the membrane. This is inside the cell. Here's the membrane. This is outside the membrane, and this particle, this small particle, can actually go in and bind to bind here, right? Once it binds, what happens is that this rod-shaped things, this molecule, actually moves. It moves down a little bit, and that little motion causes the downstream. There's another guy sitting here, another protein called Ka, which has some activity. When it moves down, once the molecule binds, this receptor, this rod, this piston, what people call the piston motion, moves down, and it blocks that activity. So the activity goes down. So as your concentration of the particle gets higher and higher, you get more and more hits, right? More and more frequent hit. So the more and more likely you have a lower activity. So the output, which is activity of the Ka molecule, which is a large protein, will actually go down as the concentration goes up. Okay? So this is a measurement. Why is it a measurement? Because from this measurement, if I get a readout of this level, I can infer what I'm seeing, right? The C is what I'm seeing if I get this readout. So if you get an input-output relationship, and that's allow you to do the measurement by looking at the output of the Ka activity. Ka is just a molecule, a large protein complex. And let's remember the measurement itself, the Ka timescale, is about 0.1 second. At 0.1 second, it can read out what happened, the C concentration. Okay, so that's the measurement time. Remember, that's the time scale. It does the measurement. Now here is what I call the nano-scale brain of E. coli, right? So if you look at the E. coli, this shape, and it has so we think about E. coli respond to chemicals as your nose respond to smell, right? Very similar. So you can think about these receptors. So on the cell, for each individual cell, you have around 15,000 of these sensors, okay? And each sensor is about 380 angstrom. That's about 38 nanometers long and 2.5 nanometers wide. That's why I call it a nanoscale brain, because each little sensor is actually about nanoscale, right? So you have a lot of them. They form this patch. So if you look under a microscope, they form this tight patch, and together they sense. That's how they can sense very, very small signal. If one particle comes in, if one sensor didn't sense it, other guy may pick it up. That's how they work cooperatively to sense very, very, very small concentration of particle. Okay, so that's the receptor, and they form this cluster, and there are all these other proteins here, chemotaxis protein here, which help to process that information. Some of which I'll tell you in the next minutes or so. Okay, so this is what I call the nanoscale brain of E. coli. Now let's go back to memory, right? We have a sensor, just like we have. We have eyes. We have nose, which have a lot of these sensors in our nose. And then to call it a brain, really, you have to have memory and processing units, right? The information we get is going to our brain and being processed. And combine that with memory, we decide, for example, if you smell something, and if that's something associated with some very bad memory, and you remember, next time you smell it, it's going to feel very unpleasant, because you recall that memory from before, and you compare with the current measurement, and you said, no, I don't like this. I'm going to move away. Okay, so that's exactly what happens in E. coli. But what's the form of memory? Right? The form of memory turns out to be also on this receptor. On this receptor molecule, you have these 4 residues, 4 amino acid residues. These 4 residues have the property that they can be modified. You can add a methyl group, CH3, or remove that methyl group by the 2 enzymes called KR and KB. So these, to me, I'm a physicist, or I know computer. I think these are nothing but bits. So you add a bit. So this is a unit, right? Quantum unit. This is a discrete unit. You just add or remove. These are bits of information. I'll tell you what it represents. So this little bob, the blue square, is a bit of information. And what the PR and KB are? Just writers and erasers, right? You write down some information, and sometimes you have to erase it, so you can write again, some other things, right? So PR and KB are the 2 molecules that are actually responsible for writing and erasing. And that's it. That's the memory. So, but still, why do you call that memory, right? Okay. And also, how does E. coli change its memory? Okay, let's first ask this question. Okay, the answer is actually it changes this methylation level or memory by something we call adaptation. So adaptation is also a very sort of universal property of living system. What do I mean by that? If you go to a very bright room from a dark place, right? So originally you just feel blind, right? Because you couldn't see anything. But after a while, your eyes adjust. You adapted to the new environment. So again, you can see, you have a contrast. You filter out the background, now you have contrast. So something happened in you that allow you to adapt to perform the same as before in different environment. That's called adaptation in the context of sensory system. Now, in the context of the E. coli, basically I have a concentration of the smell particle, right? So if I change it by a large amount, I go from 0 to a large amount let's call C, what happened to the output? Remember there's output at key A level, there's an internal output. Output will actually immediately measure this difference. I see a difference, so it will go down, right? Now, but if you observe it long enough, this activity will slowly creep back up all the way to the same level as before, even though the intensity of the smell particle is still there. So what happened? How could you have the same input and have different output here and here? So what happened is that your system itself has evolved. So if you look at the same time during this process, the receptor methylation level, whether 1 is methylated or 2 or 3, the number of methylated unit will actually also increase with time, and eventually when it's we call this state adapted. In the adapted state, the methylation level will change from a low level to a high level so that it cancels out the external signal, the effect of external signal, so therefore the activity, the output will recover, right? So basically it's a balance act. That is exactly how this memory can be achieved. So M, if you look at the adapted level of methylation, it depends exactly on the outside concentration. So this is a record of that, right? Let me give you an analogy how this actually really works. Well, sort of. The cell is a balance. Here's a balance, right? Do you know, when you go out and you buy stuff, right? How do you weigh, right? Apples, orange, peach, whatever. So you put on this side. This is something that you want to sense, right? So immediately after you put on the apple you want to weigh, or the molecule you want to smell, the balance is broken, right? That corresponds to the activity or the output suddenly dropped, so right? You tip the balance. So of course, you want to adapt. You really want the system to be balanced, right? Balance is good. So how are you going to recover the balance? You slowly add back methylation level on the receptor, on the different part of the receptor. The binding on binding will tell you the activity goes down. But the other part of the receptor can be modified, albeit with a slower rate, but eventually recover. How do you recover? You look, right? If I add 1, is it enough? No, it's still tilted. Not enough, let's add another one. Add another one until it's balanced. When it's balanced, it recovers the balanced or what we call adapted state. But this is a different state from this, right? This is actually balanced by having, on one hand, more particle you're sensing, and the other hand, more methylation group you have. So this is a balance of a different sort. But as a result of that balance, you see what happened here? I have an internal representation of the apple. It's exactly like when you go to the market, right? You put the apple here, it's still tilted, then you start put weight. Weight is not an apple, but give you the representation of the weight of the apple. Okay, so now we created, remember the definition of memory. Definition of memory is a representation of the event or the object, but it's not the object, right? So methylation level is a representation of the outside world, in this case, smell particle, number of the particle you try to smell, but it's not that. And the second requirement for memory is what? it changes slowly. Once you create this memory, you actually takes very long time for it to go away. What do I mean by long time? Long compared to what? Long compared to the measurement time itself, which is 0.1 second. And this adaptation time can be anywhere between 1 to 10 seconds, which is much longer than the 0.1 second you need to actually do the measurements. Okay, so basically what I'm saying is the methylation level of receptor measure the external attractant concentration by balance against it. The resulting methylation level at the adapted state now encodes a record or memory for the external chemical concentration it sees, right? Now you balanced this equals that in terms of if I see this, I know what's happening outside. Okay, now we have all the necessary conditions to that computation I told you before to how to compute gradient. At the previous time, I'm here. I'm staying around here, and I measure the concentration here, right? And I did measure it. And actually, not only have measured, I stayed long enough. Actually, through adaptation, I maintain a record of it, which is M, right? Now I have a representation of this guy. Now I go to a new place. This guy doesn't change very quickly, so it will still stay roughly the same, right? So there's two scenarios. If the new place have more particle, what happened? The balance is broken, right? So you still have this M, which can balance this previous 6 particle. Now this 2M is no match for the 12 particle that it put on here. So how does it know? It knows because it has this balance. Remember, this how away from the balance you are is exactly the output, right? Exactly what Ka is. So you know that. So you know, the balance is tilted towards the outside. What do? Decision is to keep running in the same direction, right? That's sorry, I put it too early. So in this scenario, your decision is, let's keep moving. It's a good direction because I'm seeing more and more. Why do I see more? Because compared to what I have seen before, this is more because the balance is broken in that way, right? Another scenario is that if I go to a place that has less particle, 3 of them, and I compare that with the M that I had before, and it's less, the balance is tilted toward the other side, right? Then I say, okay, it's getting worse. Let's tumble and change direction. So that's how using the measurement at the current time and the record of its recent past is able to compute the difference and decide whether to continue running the same direction or change direction to seek for better grounds. Okay, I think I'm done. A summary. So this is a story what people, I tell you all of this, but in the jargon, in the biology jargon, it's called chemotaxis. Its ability of many different cells, not just E. coli, other cells does that too, to be able to sense its environment, be in the chemical or temperature or pH, all kinds of external condition, so that it can use its motility. It actually moves, cell moves, to actually seek out better environment for its survival, right? So for doing that, as we said, swimming fast is not enough. It is a necessary condition, but certainly not sufficient, right? You have to be smart. You have to make the right decision. And the way to make the right decision for E. coli is that actually have a little bit of a tiny nano-scale brain, information processor, if you wish, to guide its motion. And this information processor have all these properties. It uses small nanometer scale sensors, but many of them. Each one of them may be noisy, but if you have many of them, the noise being averaged out and actually be very accurately measure the current environment. That's one necessary condition. As I told you, you also have to have a working memory. What do I mean by working memory? Working memory is something that you actually remember for long enough, then you have to forget. Because E. coli does have very limited bandwidth and capacity. You only have 4 sites. So you want to use it for something else. So you don't want to keep it forever and ever. So that's why even though you want to keep it long enough, you don't want to keep it forever. Not like our memory. Some of our memory is actually permanent. Some of it actually is working memory. So we do have both. E. coli only have one type of memory, which is working memory. E. coli compute chemical gradient to navigate towards food source. So using these two things, current environment and what happened in the past, and it moves, is able to actually compute the gradient, the spatial gradient, and so that it can navigate towards food source. I didn't tell you this, but this computation actually can be done very, very efficiently in E. coli, using very small amount of energy to do. So it's very efficient, low energy cost. I didn't talk about it. I didn't have time, but this is a fact. The last question, can we learn anything from E. coli? Maybe. All right, thank you.
Hey, we've got lots of time for questions. You're not done yet. Yes.
I just have a little question. You mentioned that it's not just E. coli, and how do you know?
Oh, okay, that's a good question. You know from many different ways, but okay, so let me repeat the question, right? I mean, the question is that how do I know there are many other cells, different other cells will have the same behavior, right? So this was done experimentally. For example, almost all type of bacteria, not just E. coli, Salmonella. Are you in the workshop or not? The answer is yes. I think it has pheromone, for example. Yeast mating. Yeast actually have to, have this ability to actually do sexual reproduction, and so actually respond to that. And there are many other things. For example, another thing, the higher thing is that neutrophil. Neutrophil does chemotaxis. But the funny is, okay, let me just continue this line of thought. But different cell.
You mean white blood cell?
White blood cell, neutrophil. Okay. But the C is, I told you the strategy of moving around and sensing different places is because E. coli is just too small, 2 micrometer, right? So because white blood cell or neutrophil is much larger, at least 10 times as large, actually was able to do that without moving. Because it's so large, it actually have sensors here, and it just compare the sensor result from the sensor by some diffusive particle that communicate between two ends. It actually was able to do that calculation without even move. Because moving for neutrophil is actually pretty expensive. So we know that by doing experiment, by put them in the gradient to see if they really indeed does what I call the biased random walk instead of random walk. We can also, in the case of bacteria, there are a number of genes that are necessary for E. coli chemotaxis, right? So you can look for similar genes, like the sensor genes, which is called MCP, right? Methyl accepting protein. And you can look at the sequence to see, they do have that too. Key A, they all have that. Key R and key B, they have the equivalent of that. So from the molecular level, from the behavior level, you can look for similarities. Of course, the final proof is actually the behavior. But you actually can look at the molecular level too in terms of understanding the origin. The origin, not only the behavior is similar, origin may be also similar, or maybe not. In the case of neutrophil, it's actually not. Okay, yeah.
What food source is it sensing? What is the binding?
Good question. Very good question. So amino acid, sugar, some bipeptide. So basically, the stuff that it needs to grow.
There's different sensors for each one?
Yes, very good question. I don't have the slides with you. E. coli also have 5 senses. But that's correspond not because of the light, hearing, and so on. 5 different type of molecule, like they have for serine, they have one dedicated receptor. For aspartate, have dedicated receptor, but also respond to maltose. But the sugar, they have a dedicated receptor called TRG, and so on. And it has an oxygen sensor, called EAR. So they have 5 of them. 5, well, just joking. 5 senses, it's just, yeah. I think, yeah.
So if neutrophils do the same thing, have the same
Yes. There's a wonderful movie you should watch that actually show that neutrophil actually chasing a bacteria. So actually, the bacteria release some signal. I mean, not involuntarily, of course, but so neutrophil was able to track that and just going through different obstacles and eventually engulf it and basically eat it, destroy it. So yes, I think there was a question at the back. Yes.
What does it do if it's in a uniform?
Ah.
If it doesn't have to move?
No, then it actually still moves because it's an opportunist. It was just moving around, searching for better environment. So you're right. So in the purely homogeneous environment, it behaves exactly the same way as no nutrient at all, as zero environment. So it only senses, remember I said, it senses the gradient. No matter the gradient is from 0 to 1, or 2 to 3, or 100 to 100 plus 2. But in the case you have 100, 100, 100 everywhere, it behaves the same as 0, 0, 0 everywhere. Yes?
I just want to be a little pragmatic. This, what you're talking about, is very interesting. How does it help us to really prevent, eradicate infection with E. coli can be very, very difficult to take care of? What can we learn from that to use in our use of prevention or therapy?
Yeah, that's a question I really don't have too much clue about. But I do know that pathogenic strain of E. coli does need this motility to be effective. So maybe we can target some of the motility genes or signaling genes to actually prevent that. But they may be. I mean, right now I think we treat antibiotics very brutal, right? Just kill the cell. But maybe that's effect. Maybe that's enough. But I tell you another story. I mean, okay, so there are people which think E. coli chemotaxis can be used. This is part fantasy, part just thinking loud. I think there are people actually trying to do some trials. That what if you actually you engineer the sensor that it senses some cancer or what chemical something released by cancer. And then also in that cell body, in that suicide bomber, E. coli, you also put in some drug. Then you can use the sensory system to zoom in to ballistic missile, to actually go in there and find the cancer cell and kill it there, locally, instead of going chemotherapy, which just kill other cells too. So that's actually good use of E. coli chemotaxis. Okay, yeah, right. So that's, yeah.
While you're answering the next question, do you think they want to see the David Rogers movie?
David Rogers movie. Oh, the new truth. Let me try to answer more questions. If there's more question, if there's no question, then we can.
No, while you're answering questions.
Oh, while okay, yeah, go ahead. Yeah. Yes, there was a question there.
On the other side of targeting cells, E. coli is a pretty ubiquitous pathogen for food supply.
Yeah.
Has anybody ever considered making something on the order of a pheromone trap by tracking things in there so they're let into their food supply and trapped and removed?
Interesting. Yeah, like a trap. So
What a pheromone trap does is an insect, we all produce pheromones, and so you can put a pheromone trap, and all the pine beetle, the males fly to the trap, and they trap them. And
that's an interesting. Yeah, but I don't know. Actually, I don't know what distinguishes pathogenic ones and not. But we do need E. coli. E. coli is not our enemy. E. coli is our friend. Normal E. coli. So if you I mean, I'm afraid if you do that, you might also trap some of the good ones and change your microbiome. You may end up having other problems.
this would be more like food supply.
Oh, food supply. You put in the.
You have contamination in a food supply.
Aha.
So how do you prevent it? Like when they had all the lettuce issues, they're able to track exactly where it comes from now. And they
Right.
tracked it to the ditch water in Yuma, Arizona. Widen ladder
Yeah, sorry, I'm glad you brought this up. I have no clue.
They're all in those classes.
Yes.
If you had this trained E. coli and it reproduces by fission, would both halves have that same training? And so in a sense it's inherited?
Good point. So you're talking about memory, right? Does the memory getting? No, because the time scale is wrong. But I think that the time scale for the methylation is long, but not that long. That doesn't last for 20 minutes. But something else can be inherited, namely the speed of adaptation. For example, the key R and key B are how fast you can write and how fast you can erase. When you divide, you give half of your key R and half your key B to your children. And if you have fast adapter, you will also have fast adapter. But you may not keep that memory. I have to think about that one. Interesting. I don't know.
Hey, we could go on.
Oh, there's a question over there. Yes, sorry.
Let's hold it down to 2 more questions.
How long did it take to do the calculation for you to know? And how long does it remember?
Very good question. Very good question.
Repeat the question.
So, how long? The question is, how long does it take to do that computation, and how long would that result last? So let me ask you. Let me answer the second question first. It will last as the adaptation. So basically, the computation will last as long as you'll be able to maintain that memory of the past. The computation is done actually almost continuously. So you're always computing, the cell is always computing the difference between now and difference between 1 second ago, right? And if you wish, that result is obtained by 0.1 second, very fast. But you're doing this continuously, so this answer keeps changing. Does that answer your question? Okay, very good, yes. They should. Oh, does IBM Watson is IBM Watson interested in this? Well, okay. Let me put it this way. So maybe not E. coli. Right? E. coli is too primitive, but we're definitely interested in the equivalent of how do you use very small, noisy, not reliable computing but cheap computing device to actually do computation, accurate computation. This is something that the E. coli shares with our brain. That we are more interested in our brain. But the one feature that's very common is that we, living system, from E. coli all the way to our human brain, was able to use rather lousy components to do very good computation. So maybe it's in the architecture, how these things are wired together, or maybe it's in the the adaptability of our little device. Maybe it's in the robustness that it doesn't go wrong. I mean, it would self-repair. All of these are very interesting general questions. Whether E. coli is the right place to study that and give insights, I don't know. But I'm also interested in neuroscience. So just in general, how information is processed in living system using lousy components, but yet enough to survive for making good decisions. Yes?
Is the piston sensor
Yeah.
of a family or unique unto itself?
Oh, it's a family.
So what else has the same structure?
Well, like I said, basically most of the receptor in E. coli, the chemoreceptor in E. coli, have this kind of structure. Whether we have that, I don't know. Our sensors are a little different. Our sensor are mostly G protein coupled receptor. These are 7 transmembrane helices and coupled to other. So they are more evolved. E. coli, at least represent the receptor I just showed you, represent a typical sensor in the bacterial kingdom. Yeah. That makes sense? Yeah.
Why don't you tell them what they're about to see?
Oh, okay. This is a white blood cell neutrophil, and this little guy, poor little guy, is being chased. Oh, okay. So,
That's not random.
yeah.
Are those bacteria?
Yeah, the little thing jumping around, bumping around. Yeah.
Is that like a duplicate?
Yes, you got it.